So welcome, everyone. I think we know most people here, but I'm Ned Kalin, and
I'm Richie.
this is Richie. And believe it or not, we've been doing this for 27 years in a row, although we actually missed last year because of COVID. So it's particularly exciting to have a real meeting with real 3D images in front of me that are breathing and talking and communicating. This is my actual first meeting scientifically, so I'm particularly excited about that. This has become a tradition at Wisconsin, this annual emotions meeting that Richie and I have been involved with in our collaboration over many years here at Wisconsin. And it really is, I think, emblematic of the work that's done here at Wisconsin from the standpoint of the interest in emotion regulation, psychopathology, resilience, and all of those things. It's a particularly exciting meeting. Richie will tell you more about sort of how it's set up and how it got developed over time in relation to the training grant that Richie has steered over the years. But just to say a few words about it, it's really fun because we have just a few outstanding speakers. We have lots of time for discussion and for interaction, which we really want to promote. And also we have travel awardees, and I'll just introduce them to you in a moment, which are here especially to learn and to get to know the speakers and also to get to know Wisconsin. The meeting really arose out of the combination of resources that we have here, many of which you're aware of, but I'll just say a little bit about them. We have the Health Emotions Research Institute, which Richie and I founded many, many years ago, at that time to focus on resilience and psychopathology and mind-body interactions, and that's still part of the psychiatry department. The psychiatry department also is very involved, as is the psychology department, and for sure the Center for Healthy Minds, which Richie will tell you more about. But all of those are part of the resources that support the work that we do, and this meeting, I think, is part of that, has become part of that culture and emblematic of that. So Richie, why don't you say a few words about how it's set up and the structure and all that, and then we can come back to more specifics.
Happy to. So yeah, it's great to be here. And actually, we did meet last year virtually. It was the year before that we skipped, the first time we skipped it. So this is an important part of our training grant on emotion research that is funded by the National Institute of Mental Health. I think we're in the 32nd year of continuous funding from this T32 training grant. And each year there is a seminar taught in the spring, and the reading list of that seminar is comprised of the papers that each of the speakers has nominated for as relevant to their talks at this meeting. So it's a great opportunity for the trainees to take a deeper dive. The trainees also are going to be leading off our discussion with questions that they've prepared for each of you. So this is really a fun and interactive part of this meeting and of their training experience. I also want to say that they'll just start off the questions, but there's plenty of time. So everyone should please have a very low threshold for jumping in and becoming engaged. So this is going to be a great group of speakers. One of the features that we have had over the years is typically the speakers are very senior distinguished investigators. And while it is true that every speaker that we have is a distinguished investigator, we have reserved a spot for a junior investigator over the years. And this year, one of our own will be speaking tomorrow, who has kind of risen up through the ranks and was a trainee herself in this program. So you'll hear more about that later. But welcome all, and I'm really looking forward to getting to know more of you over the course of the next 2 days.
Great, thanks. So we've got just a couple of slides here to run through. So one of the things that's also exciting about this year is the diversity of topics that we have. So I'm sure you've looked at the program, but it's a wide range, going all the way, as we'll start off with molecular and cellular circuit-based issues, through developmental issues, through treatment-related issues, all the way through societal issues and issues related to diversity, equity, and inclusiveness. And so we're very excited about that range. And I also think it's really cool because it sort of captures the whole panoply of where we are in psychiatry in a way, from the standpoint of trying to integrate at all these different levels. And just a few words about our speakers. Bo Lee will be starting off. I'll introduce him in a moment, talking about circuit-based research related to motivation. And Micah Bartels made the trip all the way from Europe, which we're very appreciative of. And we'll be hearing her from the standpoint of her work related to well-being. Deanna Barch will be talking about development and depression and treatment strategies, which we're excited to hear about. Sydney Hankerson, who will be coming in later today, will be talking about his really groundbreaking work related to spirituality and equity issues in relation to treatment of depression, trying to leverage work in churches actually to identify depressed individuals and to think about innovative treatments. And then, as Richie mentioned, Melissa Rosenkrantz, a member of our department and also a member of the Center for Healthy Minds, is going to be presenting some of her exciting work that's related to understanding at a brain and body level interactions related to immune function and illness that are quite relevant, I think, to the mind, obviously to the mind-body interface. So it's a really exciting group. And also want to mention that we have a lot of folks listening in virtually. I'm not sure how many, but we'll find out shortly. And want to encourage our virtual listeners to participate as well as we get into the questions and answer session. As we mentioned, the travel awardees has been also a tradition. It's our opportunity to get to know folks outside of Wisconsin and to give them an opportunity to have a really rich experience with the speakers and with our own folks at Wisconsin and to get to know us as well. Here are the names of folks. Please feel free to interact and get to know each other. A reminder, we have a poster session after this where the travel awardees, as well as students from Wisconsin, will be presenting their work. And so for the folks doing on the virtual side of things, we want you to participate. Please put your questions in the chat, and one of us, either Richie and I, will read them off, and we'll give you an opportunity to answer them. This is the poster session. I think that's at 4:30. And there's a timer. Tomorrow we've got breakout sessions. We'll talk about that tomorrow. And then finally, I wanted to acknowledge more formally the support that we've gotten, which we're incredibly appreciative of from the standpoint of the generosity of the organizations that you see here. And just as a disclaimer, or at least as a potential conflict of interest, I actually do a little consulting to Corcept, just so that you're aware of that. It has nothing to do with anything that's going to be presented today, but just wanted to disclose that. So welcome to all of you, and let's get the meeting started. So it's my pleasure to introduce Dr. Bo Lee, who has come to us from Cold Spring Harbor, and I had the opportunity of meeting him when I was at a meeting there a number of years ago and to visit Bo's laboratory. Bo is a professor there and is doing very exciting work, as you'll see, related to the most basic and fundamental aspects of emotion, motivation, fear, anxiety, reward, those types of processes using rodent models with all different types of methods, including molecular methods and genetic methods, methods to manipulate the circuits with chemogenetics and optogenetics, doing physiological experiments, both in vivo and in vitro, and really bringing all of these most modern and elegant methods that can be performed in rodent species together to try to understand the specific cells and circuits that have to do with, in general, at least as I read it, sort of appetitive and aversive types of responses and behaviors and how they interact. And obviously, this has important implications for not only understanding mechanisms, but for thinking about the development of novel treatment strategies as we think about the possibilities that we could actually in humans think about how we can manipulate specific circuits in a very relevant way in specific individuals. Bo did his undergraduate work in China at Jinging Medical College in the Chinese Academy of Sciences. He did his PhD at the University of British Columbia and then postdoc at Cold Spring Harbor and UCSD. As I mentioned, his work has been very influential from the standpoint of understanding the basic elements of the circuits that we're interested in that are fundamental to actually a lot of the other talks that we'll be hearing about. He's got numerous NIH grants, numerous publications in, as you can imagine, some of the most prestigious journals, Nature, Neuron, and so on. We're very excited to have him here and to listen to his work. So let's give him a round of applause. And we're very appreciative, Bo, that you made the trip out here to join us.
Okay, so thank you. Thank you very much. Thank Ned for the introduction. Also thank Richie and Ned for the kind of invitation and also for organizing this wonderful meeting. 27 years, it's amazing. And it's a great honor for me to be part of it, to be part of this symposium. Also, this is actually the first travel and the first in-person meeting that I am in since the pandemic started. So it's very exciting and also a little bit nervous. Yeah, so my lab has been focusing on studying neural circuits underlying behavior. We also are interested in understanding how behavior can have an impact on gene expression and how molecules can influence the function of synapse and the circuits and ultimately have an impact on behavior itself. The kind of behavior we have been focusing on belong to the category of motivated behavior, driven by either rewarding stimuli or aversive stimuli. So we are also interested in understanding some of the neural mechanisms underlying dysregulation of such behaviors, because these behaviors has been implicated in major mental disorders, including depression, anxiety disorders, drug addiction, and autism. So we have been mainly focusing on two major brain circuitries. One is the basal ganglia, the other one is the amygdala. So today I'm going to focus on the amygdala. I'm going to focus on the central amygdala here, CEA, because this structure has been studied very extensively and play a very important role in many behaviors and functions. For example, lesion of the CEA or inhibition of neurons in this structure impair learning and expression of behaviors motivated by either aversive stimuli or appetitive stimuli. Also, recent work using optogenetics show that stimulation of neurons in the CEA can in general promote such behaviors. Electrophysiological recording or imaging studies show that neurons in this structure respond to aversive or appetitive stimuli. This stimuli, we also call them unconditioned stimuli or UF. They also respond to the cues predicting such stimuli after learning. And those cues, we also call them conditioned stimuli or CF. So as you can see, the central amygdala is involved in very divergent behavior. So the question is how these structures are involved in this behavior. There are two kind of general ideas. They are not mutually exclusive. One idea is that maybe the central amygdala is a general cognitive process, such as arousal or attention. Therefore, impairment in this structure will generally impair learning or kind of behavior. Another idea is that this structure may contain distinct neuronal types, and these neuronal types may be specialized for different behaviors. So Indeed, works in the rodents show that the CA contains different populations that can be identified based on the markers that they express or the genes that they express. Showing here are two major populations marked by the gene called somatostatin in red here, or PKC delta. These are two molecules expressed by neurons in the central amygdala. The other two major populations of cells in the rodent brain in the central amygdala. So many studies, including the study from our own lab, show that these two populations of neurons indeed play different roles. This work in rodents also has been replicated and also extended by studies from non-human primates, especially the work here, down in here, from Net's lab, showing that in the monkey central amygdala, there are also these two types of neurons that in the central amygdala that express either PKC delta in green here, the somatostatin in red here, or PKC delta in green here. So largely consistent with the rodent findings in the rodent brain, but there is one interesting difference is that in the rodent, the somatostatin cell is a major population, ranging from 20% to more than 20%, depending on how you look at the markers. But in the monkey brain, the somatostatin cell actually only occupy a very small population. So what is the difference? We don't know. And what is the functional significance of this difference? We also don't know. Hopefully, we can study this difference across species and give us some understanding what does this mean. So today I'm going to focus on a study that is about somatostatin cell in the central amygdala. You may wonder why we study this, given that this finding from Net's lab that this population in monkeys is such small population. Maybe in humans also a minority. That's why we study this. The reason is that by the time we see this publication, it's already too late for us. So we started this work about 10 years ago in mice. And we found that in fair conditioning, those cells, the somatostatin cells, really play an important role. Okay, for example, when we do synapse physiology, we measure the synapse transmission to the red cell and the surrounding non-red cell. We found that fair conditioning training can potentiate the excited synapse transmission to the somatostatin cell. And we also show that if we block this potentiation, the fair memory formation is impaired, indicating that the plasticity onto this somatostatin cell is critical for fair conditioning. And this finding has been replicated by studies from our own lab and also from other labs' recent study by this paper. I want to mention that sometimes I use FFT or SOM. They both mean somatostatin. Sorry for the confusion. It's due to the different time with different terms. Then other studies also start to look at the function of this neuronal type in other behaviors. For example, people found that activation of the somatostatin cell can also drive feeding behavior. Okay, can also drive positive reinforcement. And furthermore, if you do lesion of or inhibition of the cell type, can disrupt addictive behavior. So it appears that this population, the function is very complex. Even though we can say as a genetic marker, we can label a population of cells, doesn't mean functionally this cell is homogeneous. Actually, those cells have a very heterogeneous function. So the question is, how are these neurons involved in divergent behaviors? Two very talented postdocs in the lab, Tao Yang and Kai Yu, decided to look at to address this question. So I'm going to present some unpublished work. Some of them are quite new. Actually, I haven't fully digested yet, but I just want to tell you, and I hope I can also receive some criticism or suggestions on how to further do our experiment and also interpret our data. Okay, so I want to tell you a story that those somatostatin cells encode salient event in an identity-specific and learning-dependent manner. So we did 3 types of experiments to reach this conclusion. One is to do in vivo imaging in behavioral animals. The other one is to do in vivo manipulation, in particular optogenetic inhibition during behavior. And lastly, I want to show you some data that we identify a circuit that can potentially mediate some of the functions that I'm talking about. Okay, first, want to show you some result on the imaging of somatostatin neuron in the central amygdala. So we infected these neurons with a virus expressing GCAM6 for in vivo imaging. Because this structure is very deep in the brain, we have to implant a very tiny length into the central amygdala, and then use a wide field microscope to image neuronal activity in behavioral animal and had a fixed condition. Okay, so we can give the animal different stimuli, such as a reward. When the animal is thirsty, water is rewarding. So water is kind of a reward for the animal in this situation. We can also apply tail shock or other things to the animal and see how those neurons respond to the different stimuli. So we give the animal initially 5 kind of stimuli: water, food, shock, and air puff. The reason we choose this 5 kind of stimuli is because that they induce distinct behaviors. So water will induce drinking behavior, food induces eating behavior, shock induces struggling, you had fixed mice, and air puff can trigger blinking of the eye. And then this is the heat map of neuronal activity of neurons in response to the different stimuli. And each row represents the response of one single neuron across the different stimuli. I'm going to show you such kind of data in the next few slides. So it's all organized in a similar way. Okay, each row is the activity of one neuron. You can see, based on the activity of all these neurons that we imaged to the different stimuli, they can be classified into different groups because they seem to have unique responses to each of these different stimuli. For example, there is one class of neuron that showed preferential response to shock. Okay, this is class 2 in here. So this class shows a pretty dramatic response to shock, but not to the other stimuli. And this class 3, should respond to food, not to other stimuli. And this class should preferential response to water, not to other stimuli. And there is always a class of neurons that don't care too much about anything that we present. Okay. So those neurons, they stimuli, they kind of induce very distinct behavior. So they are in different kind of modality. So the question is, what if we give 2 stimuli that can induce the same behavior? For example, if we give the animal quinine and sucrose, those are quite different liquid, but they can induce the same drinking behavior in thirsty animal. So what happened to the neuronal response? We found that even when the animal behaves similarly, the neurons respond very differently to the different stimuli. So there is one cluster of neuron respond preferentially to quinine, and there is another cluster respond only to sucrose. Okay. And what about a different behavior like eating? So we give the animal normal chow or a sweet pellet. Both can induce eating behavior. And again, we see that these neurons have a distinct response to the sweet And this class only responds to sweet stuff. And this one responds preferentially to the normal child. Okay. Maybe the difference is because of the different wavelengths, because quinine and sucrose, one is pleasant, the other one is pretty disgusting. And the sweet and the normal child also has different palatability. Maybe it's the wavelength determines the differential response in this neuron. Okay, I forgot to mention that even for this eating of the similar food, we can use the population response to decode whether in that trial the animal eats a normal child or a sweet palate. You can see the neuronal activity actually can really differentiate between the two kind of food trials, indicating that the neuron contains enough information to tell, okay, I'm eating a sweet food or a normal child. Okay. So maybe it's due to wavelengths. Then we give the animal quinine, shock, and air puff. All of these are kind of aversive, and see whether the neuron can still differentiate between the three different stimuli. We found indeed the neuron can still show a preferential response to one of these stimuli. This cluster 4, responds mainly to shock. This cluster responds to both shock and air puff. This cluster number 3. And there is another cluster show preferential response to quinine, which is here. Okay, so even for the stimuli that has a aversive wavelength, the neuron can still differentiate between those stimuli. Okay, what about the positive wavelength? So we give the animal water, food, and sucrose. Again, the neuron have response preferentially either to sucrose, to food, or because sucrose and water are quite similar for thirsty animals. The only difference is one that it is sweeter than the other. Indeed, there is a cluster that should respond to both. Okay, so there is some overlap here. And we also monitor the responses over many days. So every day we give the animal all kind of stimuli, such as water, food, and sucrose, and ask whether the representation of the stimuli are stable over time. We found that indeed, you can see across different days, the overlap and non-overlap of this neuron to the different stimuli maintained quite well over days, over several days at least. And this is true for different combination of stimuli. In this case, water, food, and sucrose. In this case, the water, sucrose, and air puff. So the representation seems to be stable. And the number of neurons response to each of these stimuli across days also remain to be stable. So similar number of neurons respond in day 1 to sucrose, and 1 week later, still the similar number of neurons respond to the same stimuli. And these are the examples of individual neurons. Cell 1 had good response to water in day 1, still have similar response to the same water in day 5. And cell 2 responds preferentially to sucrose and maintain its preference in day 5. Day 3, cell 3 to food and still remain responsive to food, and so on. Another neuron to shock. Okay. So one important thing I want to mention is those neurons in the central amygdala, they seem to care about important things, not non-important things. They don't really care too much about a sound, for example. If the sound is a normal sound, those neurons don't care about that. They don't show much response, but they are very responsive to the important things, including sucrose in this case, shock, food, those things that are critical for the animal. And those neurons care for those kind of stimuli. So probably we can say that those neurons have stable representation only to salient events over time. Okay. So then we want to look at if learning can change these responses. Okay. So we train the animal in a simple task, association task. There are two kind of trials in this task. In the reward trial, the CF sound predict that water will be delivered after a short delay. And in punishment trial, a different sound predict that a tail shock will be delivered after delay. So CF and the UF, there is a delay in between. So this is the behavior of the animal. We monitor two kinds of behavior. The leaking behavior, the animal has access to a water spot. If we have water, they can leak for water. And we also monitor the pupil size, because the pupil size is a measurement of arousal or important things happening in the environment. So we found that in the beginning, in naive animal, we present the two sound. Animals don't really care. They don't leak, don't show pupil size change. But after learning, the animals start to leak in response to the CF in reward trial, and start to show pupil size enlargement in response to the sound predicting a tail shock, okay, indicating the animal learned this task. And if we look at the neuronal response, we found that many neurons start to show response to the CF. Okay, so this neuron start to show CF response in the reward trial, and this neuron start to show CF response in the punishment trial. Remember, those neurons, they don't care about the sound before learning. So those CF responses really driven by learning. So we look at the percentage of cell respond to CF or UF in different trial types. You can see the majority of neurons respond to UF, but after learning, there's a small number of cells, 5%, start to respond to the CF in reward trial or punishment trial. And the interesting thing is, just as the response to UF, the different neurons tend to respond to different UF, they don't overlap that much. The CF responsive cells also don't overlap that much, okay? And this is the quantification to show that training increased number of cells responsive to CF in reward trial or punishment trial. There is an interesting phenomenon that is the UF responsive cell also increase due to learning. So this is something interesting I want to come back again later in the talk. Okay. If we look at the population response, okay, of the neurons, we look at this by doing some kind of dimensionality reduction based on PCA analysis, so that we can visualize the trial by trial responses of all the neurons, okay, in different trial type. You can see that in this PCA space, the trajectory of responses in punishment trial and those in reward trial are very distinct. They can separate it very well, okay? And the meaning of that, those population responses are very different in these two different trial types. And indeed, we can use these responses to decode whether this trial, the animal received the reward or punishment. Not only we can do this for the CF response, we can also do the same kind of analysis for, this is UF response. We can also do the analysis on the CF response. You can see after learning, the CF response trajectory is also distinctive across different trial types. And we can decode the future delivery of either reward or punishment in this trial. But this performance of decoding is dependent on learning, meaning that the CF response is predictive of the behavior only after learning, not before learning. Okay. So we want to look further into how learning change the responses of these neurons. So we decided to train the animal in a different task. In this case, is a go/no-go task. Okay. In the go trial, a CF predicts that sucrose will be delivered, but only if the animal leak during this decision window, and then it will get sucrose. This is a hit trial. But if the animal failed to leak during this window, nothing can happen. So no leak, no reward. In the no-go trial, a different CF will predict that quinine will be delivered, but the animal can withhold leaking in order to avoid drinking quinine. This is the correct rejection. But if the animal leaks, then it's a false alarm trial. Okay. So this is the performance of this animal you can see later during training, animals' performance pretty good, indicated by very high percentage of his trial and track rejection trial, and a low mistrial and false alarm trial. Early in training, the performance is bad. Okay. So this is just one example of animals' behavior during early training and late training. You can see early on, animal leak kind of randomly and receive quinine and water, sucrose, sorry. And late in training, the animal only leak in this GO trial to receive sucrose, but largely avoided leaking during the no-go trial, so that avoid quinine. And we found that many neurons start to show CF response in the GO trials, predicting reward. And before learning, this neuron don't show any response to the CF, okay? And some other cells start to show a CF response to the cue predicting quinine, and this response also didn't exist before learning. So one important thing here is that in this no-go trial, even after learning, this neuron only responds in the false alarm trial, but not in the correct rejection trial. So the difference between the two trials is that correct rejection trial, the animal don't receive anything, but the false alarm trial, the animal actually leak and receive quinine. Okay, so in this situation, the neuron start to respond to the CN. Okay, and this is the quantification of the percentage of cells respond to different stimuli early in training and late in training. Overall, there is an increase in the number of cells respond to the stimuli due to training. And there is a separation between the response to either sucrose or quinine. And there is an overlap between the CF response and the UF response. Okay. And again, this is a quantification to show the learning-induced increase of number of cells to the CF and the UF. This is in the GO trial, in the HIT trials, and this is the no-go trial in the false alarm trial. And again, in correct rejection trial, neurons don't care. So this just want to come back to the initial point I made. The learning-induced changes occurs not only to the CF responsive cell, but also to the UF responsive cell. So learning really increase the number of cells show response to UF, meaning that initially the neuron don't care about quinine. After learning, they start to respond to quinine. The same to sucrose. Okay, it's kind of interesting. We also did population analysis to look at the patterns of activity among all the neurons. This is the response to the CF. You can see later during training, the CF response in HIT trial kind of diverge from that in the false alarm trial and also correct rejection trial. We can quantify the distance between the population responses shown in here, indicating that training really make the responses to different stimuli to be very unsimilar to each other or have a distance from each other. And the same thing is also happening to the UF response. Learning change the UF response profile among all these cells. Okay, so it seems that learning induced changes in both CF responses and also UF responses. The CF responses, people usually think that can predict an outcome, therefore maybe can guide animals' behavior or promote or invigorate animals' behavior. So we want to look whether this is true for this neuron. So if this is true, then the central amygdala neuron response should precede or correlate with the behavior. Okay, so we look at the timing between neuronal response and the behavioral response. So here is one example neuron, the trial-by-trial responses to the sound predict a water reward. And we also, this is aligned to the onset of sound. We also put the first few leaks of the animal after the sound. So in here, you can see that the leaks occur first, followed by the neuronal response. Okay, so this is the average response for this neuron shown in blue, and average leaking behavior for this animal shown in red, in black, you can see this blue traces follow behind the black traces, indicating that the neuronal response actually is late compared to the behavioral response. We quantify this for all the neurons. Look at the delay between neuronal response and the behavioral response. Any number above 0, meaning that the neuronal response is delayed. You can see all neurons have a delayed response. Okay. We also look at the trial-by-trial correlation between the amplitude of neuronal response and also the leak rate. And for all the neurons, we don't see a correlation. And this is for the reward conditioning during training. We also look at the data for the retrieval after reward conditioning or retrieval after fear conditioning. And the result is very similar. And we also analyze data from the go-no-go task. Again, neuronal response is late compared to behavioral response. And there is no obvious correlation between neuronal response and behavioral response for the majority of cells. Okay, this is quite different from the result from a different brain area. For example, recently we did imaging in the BLA, the basolateral amygdala, using similar behavior in mice. And we found here in the basolateral amygdala, the neuronal response actually really preceded the behavioral response for this leaking behavior in reward trial, or for this running behavior in the active avoidance task. So in the BLA, neuronal response precedes the behavioral response. So it is capable of instructing the behavior. And in another brain structure, the anterior insular cortex, again, we found that neuronal response preceded the behavior response. And furthermore, many cells actually show strong correlation between the neuronal activity and the leaking rate. So in those areas, it seems like those neurons is able to instruct behavior or even invigorate behavior. But in the central amygdala, this seems to be not the case. So just to conclude this part of the talk, we found that central amygdala neurons encode salient event in an identity-specific manner. The response to salient events are dependent on learning. And the CF response seems to not able to directly drive or invigorate actions. Okay, because these neurons show very prominent response to US. And this US responses is modulated by learning. We asked whether those US responses are actually critical for learning. So to do that, we decided to inhibit specifically during US presentation of the neurons in the central amygdala by introducing a HT, which is optogenetic inhibitor into this neuron and implant optical fibers in the central amygdala. This is just to show you some examples of the histology. Those are the somatostatin neurons in the central amygdala infected with HT. This is the tract for optical fiber implantation. And the specificity of the virus is also confirmed. And we train the animal sequentially in two behavior. One is a reward-only behavior. So a sound predicts water, what a sucrose will be delivered. The animal needs to leak to get water. And then the same animal is proceeded with training in the go-no-go task. Okay. And in this task, we inhibited those neurons specifically during this US delivery time for 2 seconds. Okay. And we found that this manipulation dramatically impaired the learning in the reward-only phase, as measured by the accuracy of the animal in this behavior, and also measured by the leaking behavior after the sound presentation. Okay, so impaired the learning of reward. And then after this, we give the animal additional training sessions without laser inhibition, so that the animal eventually catch up in the RC group. They have similar performance compared to the control group. And after that, we train the animal in the go-no-go task, now in the presence of the laser inhibition. again, we found that animals' behavior is impaired by the inhibition in both goal trials and no-goal trials, as assessed by the hit rate and false alarm rate. And animals' leaking behavior is also correspondingly impaired. So basically, this experiment tells us that those neurons are essential for learning, for both reward learning and punishment learning. What about after learning? If we inhibit this neuron after learning, can we have an effect? So we give this animal additional training so that they reach similar performance for both groups. And then we inhibited those neurons during the US presentation in this well-trained animal. You can see that the inhibition really didn't have any effect, indicating that after learning, these neurons are not important anymore for these behaviors. Okay, so what about the CET response? So in a different set of animals, we train them in the reward task. After that, we inhibited during the CET presentation. We didn't see any effect. And then we proceed with the go-no-go training. We also inhibited the CET response in go-no-go training, no effect. So the CET response seems to be not critical for this behavior. So this leads us to think that maybe these neurons are really important for learning about the behavioral outcomes. And therefore, if you inhibit them during the US presentation, animals' learning is impaired. So we want to further test this idea. So we train the animal in a different behavior. It's a choice task in which the animal can freely choose to drink water in one of the two ports. Okay. But drinking in one of the ports will be coupled to a laser inhibition of those somatostatin neurons. Okay. And this laser inhibition is only happen during this port entry for a couple of seconds. And we ask whether this can affect animals' choice behavior. We found that animals really start to bias away from the port where they receive the inhibition, as if they don't feel that they are drinking water there. That's our interpretation. And therefore, they start to choose the other port for water. So we think this result is consistent with the idea that those neurons are important for outcome evaluation. Just to summarize this part of the talk, we found that the US responses are essential for both reward and aversive learning, and the US responses probably are needed for outcome evaluation. Then the last set of data I want to show you is about how these neurons actually mediate this behavioral effect. That is from which pathway, which circuit. Okay. When we look at the projections from the somatostatin neuron, there is one projection that is quite prominent. That is to the dopamine area in the midbrain, especially the SNC. And so this is revealed by anterior grade tracing. The neurons are infected with a tracer, and we follow the axons. You can see these axons in the FNC. And we confirmed this using retrograde tracing in these mice in which somatostatin neurons are marked by a reporter, which is a nuclear localized GFP. You can see the central amygdala filled with those somatostatin cells. And if we inject CTB into the FNC, we'll label a lot of cells in the central amygdala, and almost all of them are somatostatin cells. So the somatostatin cell constitute basically probably 100% of this projection pathway. And this pathway is particularly interesting to us because we know that dopamine cells are important for learning. Okay. So could this pathway be critical? So we decided to specifically inhibit the terminals of this projection during learning. And again, we trained the animal in the same task, basically reward task only first, followed by go/no-go training. And we inhibit these terminals only during the presentation of the US in both cases. Okay, so we found that in the reward only phase, animals' behavior is really impaired. Okay, indicating that this terminal or this projection is critical for learning. And however, the go/no-go task is not affected by this infection, by this inhibition. Seems like the punishment or aversive learning is intact if we inhibit this pathway. And we confirm this by training the same animal in a fair conditioning task in which we inhibited this terminal during the shock presentation. And we found that that didn't really affect the phrasing behavior during retrieval in the next day. So indicating that the fair learning is not affected. If there is any effect, it is that the phrasing behavior during the training day actually is slightly potentiated by this inhibition. So we concluded that this pathway is specifically required for reward learning, but not for punishment learning. So the question is how this pathway regulates reward learning. Obviously, dopamine cell is a candidate. So can this neuron in the central amygdala affect or modulate function of dopamine cell? To directly address this question, this actually has been hypothesized in the literature for a long time that the central amygdala may modulate or somehow regulate dopamine cell in the midbrain. But so far, we don't have direct evidence for that. So to address this question, we use a double transgenic animal, somatostatin flip and that pre, so that we can infect somatostatin cell in the central amygdala with the channel rhodopsin and infect dopamine neuron in the FNC with GCAM6. So we use light to stimulate neurons in here and use photometry to measure dopamine neuron activity in here in the same animal. And this is the data. And here is the heat map of trial by trial response of the dopamine neuron measured in here in this animal in response to light. Okay, a warm color meaning increase in activity. You can see that light really increase activity of dopamine neuron in the FNC. We also simultaneously monitor animals' behavior. Animals have access to a tube to leak for water. And this manipulation doesn't affect animals' leaking behavior. Okay, this is how the average trees look like. Upon laser stimulation, there is a quick physical activation of dopamine neurons in the FNC, and this laser doesn't evoke any leaking response. This is the quantification of all the animals. And in the same animal, we can also give the animal water. And we know water can induce dopamine neuron activity. Indeed, so water reward induce a physical dopamine neuron activity in the same location as in here. And in this case, this animal actually leak because they want to drink water. So this dopamine activity actually correlates with the leaking behavior in this animal. Okay. And we repeated this experiment in the VTA. Okay, so do the same experiment and measure dopamine activity in the VTA. So we basically see the same thing. Activation of somatostatin neuron central amygdala induce a physical activation of dopamine neuron in the VTA, and no effect on leaking behavior. And if we give the animal reward, also induce dopamine activity in the VTA, and that correlates with leaking behavior in this animal. And in a control experiment in which the central amygdala don't have channel rhodopsin, but instead have GFP, we do laser stimulation, nothing happened to dopamine neuron. Okay, but this animal, they do have a dopamine response if we give the animal water reward. So indicating that our stimulation induced dopamine activity is actually due to activation of somatostatin neuron, not just because of the light effect. Okay, so activation of those neurons is sufficient to activate the dopamine neuron. You may say, oh, this is artifact, normally it doesn't happen. So the question is, whether normal or natural dopamine activity require or depends on somatostatic neuron in the central amygdala. So we need to inhibit this neuron in the natural behavior, say dopamine activity is affected. So to inhibit this neuron, we use a chemogenetic approach, and to image, we use grain length in the dopamine area to image single cell activity in dopamine area. Okay. So for the chemogenetic, we use cord. The ligand of cord is called cell B. Okay. So this is one example of dopamine neuron activity trial by trial in the VTA. Before cell B injection and after cell B injection, the same neuron. You can see before there is a high activity in response to water. So the animal is naive animal, we simply give water. So every trial you have a response. But after we inhibit the central amygdala neuron, the activity becomes decreased. And this is the quantification of the same neuron, average response before inhibition and after inhibition. We did a control experiment because cell B used DMSO to dissolve the vehicle. We repeated this experiment by simply inject DMSO. We look at the response before and after the injection, no change in dopamine activity. So this change is truly dependent on inhibition of this cell. It's not because we just repeatedly image the dopamine neuron. Okay. And this is quantification of all the neurons. There is a general reduction of activity if we inhibit central amygdala. And for each animal, it's consistent. There is a reduction of activity in dopamine activation due to inhibition of neurons in the central amygdala. So this is naive animal. What about learned activity in the dopamine area? We know dopamine neuron, they are famous for predicting reward prediction error in coding. These things, we want to look at that. So we train the animal with a task, a simple task, a sound predicts reward. So we can image dopamine neuron in response to the CS and the US. And we can also look at the unexpected delivery of reward to see if those responses also depend on central amygdala. Okay. And this is one example neuron. You can see that inhibition of central nucleus decreased activity of the neuron to the CS and also to the US. So in a lot of neurons, the CS response and the US response of VTA neuron are inhibited by inhibition of central amygdala. Okay. So again, we did a control experiment, and DMSO treatment has no effect. So it's not due to repeated imaging. Okay. So if we look at all the neurons, we found that inhibition of the central amygdala in general decreased the CS response and also the expected US response. Okay, consistently. Interestingly, some neuron, the response to the unexpected delivery of US is differentially affected by inhibition of the central amygdala. Some neurons show increased activity, some neurons show decreased activity. On average, there's no change. It's a very interesting distinction between the unexpected reward and expected reward, meaning that the error encoding of dopamine neuron dependent on the central amygdala. Okay. This is a control experiment showing no effect for DMSO. Okay, I think I spent a lot of time already. Just to summarize this part of the talk, so we found that the US responses of those projecting to the FNC are specifically required for reward learning, and activation of these neurons is sufficient to promote dopamine neuron activity in the FNC and VTA, and the activity of these neurons is also required for normal dopamine function. And this is just full list of the summary, but the take-home message is that we found the somatostatin cells in the CEA send the projection to the dopamine area, and this projection is critical for normal dopamine function. And we think that probably underlies the function of this pathway in reward learning. And with that, I want to thank the people who actually did work. So this, the current member in the lab, and as I mentioned, Tao Yang and Kai Yu are the two brilliant postdocs who really in charge of this project. I have a lot of collaborators, and also thanks for the funding agencies. Thank you all for your attention. So much, Danielle.
Okay, hello everybody. I'm Danielle. I am a graduate student and in the neuroscience training program. And I'm part of the emotion research grant. So we spent a lot of time this semester reading the works of the presenters today and prepared some questions for them. So my first question for you is sort of a broad, big picture question. Well, first of all, thank you for your talk. It was a really great talk about some really impactful material. So thank you for being here.
Thank you.
Yeah, so my first question, broad, big picture question is, how do you think this information will apply to human work, such as for drug development for anxiety-related disorders, fear-related disorders like PTSD, phobias, and anxiety?
Yeah, that's a great question. Also a challenging question to address. As I mentioned in the beginning, so although work from rodents is fundamental and insightful for understanding maybe similar processes in human brain, but there is a gap between rodent research and human research, and even between rodent research and non-human primate research. So the work in here by Ned's lab already showed that in the non-human primate, it seems like the cellular composition is different from that in the rodents. So I can speculate what would be the functional significance of that. So first of all, fewer number of neurons doesn't necessarily mean that those neurons are not important. First of all, those neurons, the somatostatic neuron I'm talking about in the non-human primate, could still carry out similar functions. Or during evolution, there is another type of neuron, or those neurons just lose their marker, but there is a type of neuron that is marked by some other genetic markers that perform the same function compared to neurons in the mice. So those the neurons that I characterize in here, those are the fundamental processes in response to either reward or punishment. From there, we can start to think how those neurons in monkeys or humans respond to similar stimuli, and what's the connection from those response to dysfunctional structure, dysfunctional circuit function, and also how that can lead to psychiatric disorders. We know that a lot of mental disorders, although in humans, manifest as high order emotional or cognitive functional deficit, but they also have fundamental dysfunctions in perceptions of the world, in feeling of reward or punishment, in eating behavior, feeding behavior, or autonomous behavior, autonomous function of the nervous system. So those brain-body interactions can have a profound impact on human health. I think we are in a position to start to understand from a simple rodent behavior, try to to get some insight of how higher cognitive function can be linked to those perturbations.
Okay. Another question that I wanted to ask you about was related more to the somatostatin negative cells. And this wasn't actually, we read a lot about this in your work, but it wasn't actually something that you talked about a lot in the talk. So as an intro to this question, I was wondering if you could maybe mention some of the roles of the somatostatin negative cells in the central amygdala.
Yeah, that's another great question. As mentioned, so the Pikachu data cell is probably the major somatostatin negative cells in the central amygdala. And a lot of study in rodents clearly shows that those neurons have different functions. So we found that more than 50% of these neurons, they are preferentially activated by foot shock in fair conditioning. And research from other labs, including the research from the WSoE lab, they found that activation of these neurons have a anorexic function, suppressing feeding behavior. Those neurons, we found that also send projection to kind of different brain areas compared to the somatostatin cell. So one major difference in the connections may be that the Pikachu data cell has a very strong projection to the adjacent central medial amygdala, the CEM. The somatostatin cells seem to have more long-range projections, including the FNC projection, the midbrain projection, and also to peribrachial nucleus, to PAG, even to the hindbrain, for example, the NTF. So those seem to involve mainly the somatostatin cell, not the Pikachu data. Pikachu data cells still project to that, but minor. They have, on the other hand, a very strong projection to the CEM. Maybe they do function as traditionally predicted for the CEL. CEL has been predicted to or hypothesized to disinhibit neurons in the CEM, and therefore, which is a major output of the central nucleus of amygdala, therefore have a effect on the final output. So the Pikachu data cell may function in that role. It definitely has a function in fair conditioning and also maybe also in controlling feeding behavior, but there has been no extensive study in terms of characterizing the encoding properties of the Pikachu data cell. Mm-hmm. So the only functional evidence is through optogenetic activation. As you know, when we do optogenetics, we simultaneously activate all the neurons synchronously, and you probably will see a behavior output that may not necessarily represent the natural function of those neurons. So we need to interpret those data with care. We did some imaging study of Pikachu data cell only in fair conditioning. You know, we didn't do anything else, although we are doing this right now, because we start to realize from the work on somatostatin cell that maybe we need to characterize the responses in the same animal to different stimuli, not only focus on fair conditioning. Because initially we only image the somatostatin cell only in the fair conditioning. But when we start to image other stimuli, we start to see a different world. That is, they are not specialized for only fair conditioning. The central amygdala is not specialized for only fair conditioning. The reason people talk about fair conditioning so much is because traditionally we do a lot of fair conditioning. We don't do much else in rodents. Right now, we start to be able to do many things. Even in rodent, we can design complex tasks, including cognitive tasks that has not been possible for rodent work. So right now we start to see, okay, those neurons may have so-called mixed encoding. So one neuron can do many things, and then a population of neurons in combination, they can really encode a very complex stimuli in the whole world to give the animal the perception of what is really going on. Not to say that, oh, this neuron is only in charge of fair conditioning. I think that's a misleading thing in the field. Okay.
Let's open it up for questions.
Got two microphones over here, so
Yeah, come to the
why don't you grab a microphone? I'm going to go in the back and see what we have in the chat.
Thank you for some really elegant studies. It's really interesting work. I was wondering, because you mentioned that earlier on in your talk that these cells didn't respond to sound. And I was wondering, I have a second question, depending on what was that sound that they were being exposed to? Was it a tone or was it
Oh,
a?
yeah, so we use mostly the tone. For
Okay.
example, 4 kilohertz tone, 12 kilohertz tone. Yeah, the tone response regularly between 70 to 75 dB. Yeah.
So I was wondering, because it seems like throughout your talk, you suggested these are really important for, somewhat survival skills, food, water, those kinds of things. So I was wondering if you expect there would be a difference if you expose that animal to maybe a predatory noise or something like that.
Yeah, that's a great question. Indeed, we didn't do that, but I predict if we do so, those neurons would respond to that. Yeah, those are naturally important for the animal to survive. Basically belong to the category of salient events. I think those neurons should massively respond to that. Yeah, we should try that.
Bo, if you don't mind, let me go ahead and ask a question from the virtual audience. We'll go back and forth. This actually is from Rotem Kovner, who is the postdoc now whose work you quoted from the standpoint of the somatostatin work in our laboratory. So she's at Yale now. And Rotem, it's great to have you with us. We wish you could be here in person.
Thank you.
But her question is, Dr. Lee, wonderful talk and very interesting data. Here you are looking at the function of the somatostatin cells and their responses to various stimuli. I was wondering if you knew or are looking at the function of the somatostatin protein in these behaviors and responses, considering that the central nucleus is composed primarily of inhibitory neurons.
Yeah, that's a great question. So far, we have been using somatostatin only as a marker. It's kind of a shame that we've been doing this for many years, just to use a marker to mark a population of GABAergic cell in the central amygdala. And we only talking about those neuron-released GABA. We completely ignore the fact that they also release somatostatin, which is a very important neuropeptide. We haven't really have any data yet with regard to the function of the peptide itself. But we are aware of that. We started to work on that. We're actually collaborating with a lab in China, Peking University, Yulong Li, who developed a sensor for somatostatin. We actually right now can start to measure the release of somatostatin. I think the sensor works well. And the question is how those, when somatostatin is released during behavior, and what is the impact of somatostatin in addition to the fact that this neuron also releases GABA. But the hypothesis is that the peptide, neuropeptide, maybe have a slower effect, even though they are released maybe quickly, but the effect going to be slower than GABA, maybe on the order of minutes instead of milliseconds. But the effect can be long-lasting because the neuropeptide may work on those receptors, changes into cellular signaling and genetic programming. So very exciting area, but we don't have much data yet. So we are working on that. Great question.
Yeah.
Thanks so much for your talk. Very fascinating work.
Thank you.
One thing that I found particularly interesting was that these somatostatin plus neurons are primarily responsive to very salient stimuli, conditioned stimuli, and unconditioned. And it made me think about fear extinction because, as you know, I'm sure it's learning the absence of a salient stimulus. So I don't know if any work's been done already, or if you would speculate as to whether it would be likely that these neurons had an important role in fear extinction, or perhaps that we'd have to look at other neurons.
Thank you very much. Actually, thank you for this question. I do have some slides that is related to your question. We look at the responses of this neuron during extinction in either the reward trial, the reward conditioning or fear conditioning. You can see this is the recall session. The behavior in recall in extinction, meaning that we play sound after learning, but don't give the animal water. So initially the animal leak in response to sound, but gradually they stop leaking because they realize they don't have water. So this is the quantification of leaking in the first 5 trials and the last 5 trials. If we look at the neuronal response, it's very interesting. There are 2 populations of neuron. One population, they show response in the first few trials, but then they don't have response anymore. when the animal realized there's no water. So this is the first fall trial, the late fall trial. But there is another population. They don't have a response in the beginning, but then start to show response when the animal behaviorally extinguished. Okay, so this may be corresponding to the extinction neuron. Okay, because they show response specific after extinction. And what happened? So in the fair conditioning, we found a similar neuron. Basically, here we measure the eye blink here. So after fair conditioning, we place on animal, close the eye. Okay, this is one convenient measurement we have. In the first few trials, the animal close the eye, and then they don't care anymore after extinction. And this behavior, again, there are two populations of neurons. One population show response to the sound in the first few trials, the blink an eye. But then there is another population that only show response later on. These neurons eventually don't respond, but these neurons start to show response after extinction. So there are extinction neurons belong to those somatostatin cell. And it seems interesting that the extinction neuron for fair conditioning and the extinction neuron for reward conditioning are different. Okay. So previously, people know that central amygdala is critical for extinction because people record this extinction signal, as we're showing here, and they inhibit the central amygdala. Animal has impairment in extinction learning. The difference we see in here is that we actually see the different population in charge of different extinction learning. So that's the new thing we found. So far, we cannot specifically inhibit those neurons because we don't have a way to do that. We can only inhibit the somatostatin cell. That we know that they inhibit extinction learning as well. So we think there are extinction-specific neurons belong to the somatostatin cell. I think new tools and new way need to be developed in order to specifically target those neurons. Excellent question.
Thanks so much. That's extremely interesting. And if it's okay, I might ask a quick follow-up question and then I'll let somebody else take the mic. Just what you said about having extinction-specific neurons and even earlier about specific cells for certain functions. And you mentioned that we can't currently isolate them. Do you know if anyone has done any work or perhaps you have looking at whether they might in some way be biologically distinguishable and perhaps in the future targetable?
Yeah, so I think those specific type of neuron, they definitely should be distinct in terms of the connectivity. Otherwise, they wouldn't specifically respond to different things, right? They should receive different input, therefore they have distinct response. I also hypothesize that they may project to a certain location or at least even to the same location, may be different target neuron. So they are wired to be differently, I think. So one way to do this, to verify this, maybe we use some kind of way to tag those neurons during our imaging. If there is a magic thing, molecule that we use a laser to shine on that particular neuron bone, we label this neuron. And afterwards, we look at the connectivity, input and output. Maybe that's one way to do it. And also maybe look at this, the RNA expression, profile, the whole gene expression profile, maybe to identify some molecule that can specifically target those neurons and eventually study that. I do believe that they should be different.
Very fascinating. Thank you.
Go ahead over there.
Very nice talk. I just had a couple of quick questions. So first, in response to this new data right here, one thing you might want to consider is using the TRAP2 system, because if you could just trap it in one state versus the other, you might be able to selectively manipulate it. The questions that I had were, one, it's really neat that these neurons are clearly encoding salience and not valence. So do you think that there are distinct projections, for example, that are encoding the aversive stimuli, maybe to the RMTG or peribrachial nucleus, that are responsible for that? Because clearly that's not the VTA projection. And then the second question I had was the seemingly parallel effect when targeting the VTA versus substantial niagara pathway. Do you have any evidence to see whether that's a collateralization of the same neurons or are those distinctly different neurons?
Yeah, that's all great questions. So the first question maybe whether the aversive neurons and projection to some specific locations, RMTG or other location. We haven't looked at RMTG, but we did find some interesting projections. We published a Journal of Neuroscience paper last year or 2 years ago. So we found a projection from the somatostatin cell to the GPE actually. And if we inhibit those terminal that impair fear conditioning, I have to admit that we didn't do reward conditioning. Although it impair fear conditioning, we cannot claim that it is specific for aversive learning so far. So we are following up on that to see if that pathway is distinct from the pathway we see in here. And there are indeed other pathways. This the peribrachial nucleus pathway. It is an interesting pathway because we know based on Richard Palmer's work, this peribrachial nucleus to central amygdala really convey this aversive UAT information during fear conditioning. And they also receive feedback from the central nucleus. Maybe this pathway can also potentially be involved in the fear learning. So those are excellent questions. We need to look at that. With regard to, what is the second question again?
Collateralization.
Yeah, collateralization to the FNC or VTA. This is kind of a challenging question to address because so our hypothesis right now is that, as you know, the somatostatin cells are GABAergic. When we do stimulation, you increase the dopamine neuron activity. The hypothesis is this is due to disinhibition. So we know that we have data. I don't have a slide to show you, but I have data to show that those somatostatin cells really preferentially, 90% of them innervate GABAergic cell in the VTA and FNC. Only 10% or less directly innervate dopamine neuron. Okay, and also we know that the GABAergic cell within those midbrain area, they actually can be long range, meaning that those neurons, the GABAergic neuron in FNC, can send a projection to VTA to innervate dopamine neuron in there instead of locally. So those virus or CTB can also not easily distinguish fibers of passant versus the synaptic terminal. If we inject either virus or a tracer into the FNC, we may as well contaminate the axons to the VTA. So it's kind of hard. So we are trying to use different ways and selectively inhibit terminal in the FNC water VTA is also not that easy, but we are trying to use a refined way for optogenetic stimulation. That is at a single cell resolution using holographic stimulation and imaging simultaneously. So maybe in that way, we can assure that we activate one axon terminal or one neuron and look at the effect on the local area in the imaging area or some another area. So we are trying to do that, but it's slow. It's not that easy.
So let me ask a question from back here from the virtual audience. This is from Cecilia Hillard. And she asks, what do you think is the mechanism by which neurons become recruited into the response positive ensemble?
The mechanisms. Okay, so there are, well, one of the mechanism is through a synaptic plasticity, LTP-like phenomenon that we described about 10 years ago during fear conditioning. There is really a synaptic potentiation onto neurons in the central amygdala, especially the somatostatin cell has very potent synaptic potentiation. And those could increase the response of this neuron to initially neutral stimuli after learning. And there is one interesting thing that is here I presented data that not only the CF responsive neuron increase, the number of US responsive neuron also increase. Okay, and when we do slash physiology, we found the same thing. Basically, if we give repeated training, there are massive increase in synaptic transmission from different stimuli, different source, not only the input from auditory area, but also the input from the parabrachial nucleus, for example, that's supposed to carry those salient information. And those responses also increase. I think synaptic changes could be at least one of the reasons why learning and change or recruit different additional populations into the ensemble.
Let me just remind folks, when you ask a question, if you would introduce yourselves and just say where you're from, that would be a nice feature. Go ahead.
Hi, I'm Zach. I'm a grad student at Northwestern. I really enjoyed the talk and was curious about the subpopulations of neurons that were responding to different types of stimuli, some all within the central amygdala. Yes, you kind of showed functional specificity of different populations. And I was wondering, was there also spatial, are they spatially segregated? And is that spatial localization consistent between mice? Are they all the same population, is always in the same space?
Yeah, that's a great question. So if the first question is those non-responsive cell, those silent cell that you're interested in, or yeah. So yeah, in all those experiments, we always see some cell don't respond, but we only give a limited number of stimuli, right? And 5 or 6 sometimes. I think maybe those neurons are reserved for additional stimuli. This is the one speculation. And also maybe there is some kind of instability, although in general, there is a stable representation of different stimuli over time, but there's still some drift maybe. Some of the silent neuron in one session may be recruited to the active ensemble in some other session, although I think this is a minority, but it can still happen. The more likely feature or possibility is that those neurons may be responsive to some other stimuli that we didn't include in here. But I think those stimuli should be salient, important for survival for the animal. This is the general feature of this neuron. In terms of spatial clustering, we did indeed see some spatial clustering, although it's not clear. It's not like in one spot you have a responsive cell to sucrose, another spot to pine. In general, they are intermixed, like peptide sauce. But if you do detailed analysis, there is some kind of pattern. So for example, those response to shock seem to be more scattered in the whole field of view. And those response to the water, for example, seem to be more clustered in the center in the view, in field of view, in one location of the field of view, for example. So there is some general distinction of the spatial clustering. We still don't understand what is the principle in there. Thank you. Yeah.
Hi, thanks for the talk. A really thoughtful progression of experiments. One thing that was really interesting was the kind of lack of a trial by trial correlation between the neuronal activity and the behavior. So I was just wondering if you could talk a little bit more about why you think that might be, that there's not necessarily a trial by trial relationship, but maybe a task wide or condition wide relationship. And if you've considered maybe any kind of lagged correlation or lagged analysis where potentially activity could be related to behavior on the next trial or something like that.
Yeah, that's an excellent question. So we did do analysis to look at whether the previous trial has an impact on the next trial, either do this strict one trial, next trial correlation kind of analysis, or a volume basis, correlation analysis. We don't see any obvious correlation for the vast majority of neurons. As I mentioned, in some other brain, like the BLA, and in the insular cortex, we do see very strong correlation. Many cells correlate with that. So we don't think this is an artifact of our analysis or imaging. I think this truly represent the properties of this neuron. So the question is why? Why this neuron behave like this? So in general, I think if we want to see a neuron is directly linked to a particular behavior, the neuron activity should precede and correlate with the behavior. And this neuron don't. We think those neurons probably, they are not directly related to the ongoing action of the animal, but it will tell the animal the outcome of a particular behavior. Okay, so basically when the outcome is revealed, whether it is aversive or rewarding, And this is the moment those cells really care about. And that's tell the animal, okay, this is a good or bad consequence. So this is outcome evaluation that has a profound effect on learning per se. It may not necessarily tells you exactly what you need to do in this particular moment, but you need to form a association between this event and in general of your behavior. So I think I tend to think the central amygdala tell the animal that something is going on in the environment. You need to do something, but what to do and how to do it, I don't know. It's not my job. I just tell you something is important.
Thank you so much.
Hi, my name is Lana Grasser. Thank you for your talk. Oh, yes, of course. Hi, I'm Lana. I'm just graduated from Wayne State in Detroit. I was curious, and I think this dovetails nicely with the speaker from the chat last question.
Yeah.
If you saw, or if others have looked at any role of early life experiences in the quantification of these cell types. So for example, if they've experienced any sort of maternal neglect or other sort of environmental deprivation.
Yeah, that's an excellent question. We didn't look at that, but it's definitely an important question to look at. I can speculate. I think because experience, learning experience has a profound effect on the responsiveness of this neuron. I think any early life learning experience would have a long-lasting effect on this neuron. Okay, but it's not simply a encounter of some random events. For example, if we just give the animal water, food, and without training the animal, just give them and imaging over time, the representation is stable. But when we couple a sound with food or water, so the animal needs to do something in reaction to the sound. So basically, the animal needs to actively use the experience to do something on their behavior. When this happens, those neurons become modified. It's experience dependent on learning dependent. So if there is an important early life event that make impact on the behavior of the individual, I think those neurons are going to be modified. Yeah.
Thank you.
Hi, Deanna Barch, Washington University. You stole one of my questions. Excellent question. But maybe related to that, I'm wondering if you know more about the normative kind of developmental trajectories of the functionality of these neurons and circuits, right? Like, are they wired up and operating in the same way all across the course of development, or do you know anything about sort of how that happens?
Yeah, that's an excellent question. And I don't know much about the development of this circuit. One reason for that is my knowledge is kind of limited on the developmental side. Another, actually, maybe I can blame that, is a general issue is the developmental study in the brain has been heavily focused on cortical areas and maybe some of the subcortical area, but central amygdala has been understudied. We know those neurons has, in the very beginning, has the same origin as some other GABAergic neurons. For example, DHL6 or something, is the master regulator of the development, specify this GABAergic cell, and its origin. The origin of the cell is kind of similar to the GABAergic interneuron in the cortex, but later on those neurons become very different. When they are in the location, they become different from the GABAergic neuron in the cortex, which are mainly interneuron. But those neurons, the somatostatin cell, they actually are not interneuron, they are projection cell. The only analogy I can have is that they have similarity to the medium spanning neurons in the dorsal striatum, maybe nucleus accumbens, I don't know, but at least dorsal striatum. And Larry Swanson made a very good analogy during evolution and also during development that maybe the BLA and the central amygdala is another set of cortical striatal system similar to the real cortex and striatum. The difference is that the cortex had different layers, more developed, and the striatum had more complex or well understood circuit, wiring diagram, direct pathway, indirect pathway. But in the BLA, we don't have a layer, we only have LA, BA. In the central amygdala, we essentially don't know much about the details of the developmental trajectory, and the wiring diagram is also less understood. Although we know there is a general disinhibition between the CEL and the CEM, but in terms of neuronal type and the details, we don't know much about. I think a development study on this circuit can give us a very good handle on one hand to manipulate the circuit, and on the other hand, how early life experience or disease-related condition mutations can have an impact on the circuit. And I think this is exciting, although it's less understudied, but it's an exciting period that we actually can do something about it right now.
Thank you.
Hi, I'm Natalie Pilgrim. I'm coming from Emory and the Yerkes Primate Center. So I was especially really fascinated by these possible cross-species differences. And I guess my question is two-part. One, do you think maybe there are if these neurons would be modality specific, comparing an aversive sound versus an aversive smell? Maybe that would have implications for species, since rodents are with mice being more olfactory versus monkeys being more visual. And then also just kind of a general clarification question on the neuroscience. The 6% versus 20% difference, is that significant in general? I don't know if there are general principles about how much it matters to function, how the population, the percentage of a population of neurons that are of a particular type. And if you could speak to that and explain that to me a little bit more.
Yeah, so that's a great thinking that different species tend to predominantly use one sensory modality versus the other. Whether the difference in the number of cells in smell studying cell in the central amygdala between monkey and mice is due to that could be, I don't know. So we didn't, so far all our study has been either using light or using sound. We didn't use smell. Whether smell can recruit more neuronal responses, compare a neutral smell or a odor from co-species or a predator would be different. These are excellent questions we didn't study. I think indeed the change in behavior, the change in behavior pattern, the change in reliability on different sensory input definitely can have an input on how the neurons are wired and the neuronal population, whether one is dominant versus the other. 20% difference or 20% for one population, the other one is only 5%. Whether this is important to differentiate between mice and the monkey, I don't know. As I mentioned, we only look at one marker. The smell study is a very convenient marker. But my thinking is we need to more rely on the functional definition instead of using this marker, because in the end, how the neurons encode different stimuli and how these neurons respond to the stimuli and then control behavior is the key thing that we are care about. If there is another marker, let's say a different marker in monkey, and the marker a population of neuron is exactly the same as the smell study neuron that I talk about in here. I mean, that is the most important thing. The actual marker is going to only give us a tool for me right now. Although we also study the smell study gene, the smell study as a peptide. If we can reveal the peptide itself, the function of that, maybe that's going to be important. That if there is reduction in neuropeptide smell study in monkey, let's say in the central amygdala, what is the functional significance of that? That would be a very interesting question.
Really interesting. Thank you.
Can you put that on?
Danielle has another question. So far.
I in fact have many more questions. So I had another question about the PCK neurons in the central amygdala. While reading several of your papers, I noticed that in your 2017 paper in which you delineated the connections between the central amygdala to the lateral amygdala, the PCK cells drove aversive learning. But in your 2013 paper in which you were delineating the central media lateral to the central medial circuit, the PCK neurons were inhibiting the somatostatin neurons, which in turn inhibited aversive learning. So in one study, the PCK neurons drove aversive learning, and in one study they inhibited them. And so I was wondering if you have any ideas on what might be the cause of these contradictory effects in this neuron population, if it might be mixed encoding of the PCK cells, or if perhaps there are subpopulations of those neurons that have yet to be categorized.
Yeah, that's an excellent question. So I'll give you guys these papers to read, and also revealed a lot of issues with our study, I guess. So I try to address those issues. So in the very beginning, when we started this study, we have a very simple-minded, idea, because we thought, field conditional is a simple behavior task, and the circuits maybe look very simple. We have two different populations seem to work oppositely, increase or decrease activity in response to some stimuli. Then we came up with this simple-minded diagram showing, there is fear on neuron, fear off neuron, and when one is excited, the other one will be inhibited, and then the output would be freezing behavior. But as the study goes on, it turns out that the simple behavior is not that simple, and this circuit becomes very complex. As revealed, even this somatostatin cell is so heterogeneous functionally, and the PKC data cell has been, less studied in terms of the coding property. We are working on that right now, and the preliminary data show that they are as heterogeneous as somatostatin cell, in response to different stimuli. So the initial model is too overly simplified. We only consider two populations, excited and inhibited. So right now, I think there is more functional domains within the circuit. That is, there is definitely we know that there is interaction, the mutual inhibition between somatostatin cell and PKC data cell. That is correct. And there is also mutual inhibition among each of these classes. Somatostatin cell can also inhibit another somatostatin cell, PKC data cell can do the same. So depending on the moment, depending on the experience of the animal as well. So maybe, the interaction is dynamic. When in a certain situation, you're going to have some, inhibit neuron, disinhibit neuron. And if the situation is aversive or punishment learning, then the net output would be driving to some output leading to a avoidance response or freezing response. And in appetitive learning, it's going to be different. So it's not going to be a static interaction among two population of cells. I think the structure is designed that the dynamic interaction among so many finite circuits within the structure can ultimately regulate behavior. I think it's still valid that PKC data cells, somatostatin cell, they are distinct. They serve different things in terms of encoding, and then they interact with each other, and then coordinate the behavior output, and they play different roles because essentially they have different connections, in terms of the output and the input as well. The input also different. So we need to revise those simple-minded diagram into a more realistic network model to try to, account for the functional heterogeneity and also the behavior, specific behaviors they might serve. I think there's one thing I want to mention. The general theme is somatostatin cell and PKC data cell tend to disinhibit some neuromodulatory system. In the somatostatin cell, it seems to be the dopamine. The PKC data cell, they can also do the same thing, but they may regulate either a different population of dopamine cell. We know dopamine cells themselves are heterogeneous. Some of them in charge of reward, some of them actually respond to punishment or saliency. But the PKC data cell can potentially also regulate the cholinergic system, and therefore have an impact on learning in the BLA. So basically, seems like the output of the central amygdala, at least one major function is coordinate the neuromodulatory system, and eventually have an impact on learning.
I've got a question for you, Bo.
Yeah.
We're particularly, as you know, we're particularly interested in temperamental differences. And I was struck by the stability that you were showing within an animal from the standpoint of the responsiveness of the salience, whether it was positive or negative in the somatostatin neurons. I'm just wondering, have you observed individual differences across animals in the numbers of neurons or the intensity of responses to reward versus punishment types of stimuli?
So yeah, so we imaged typically 6-7 animals. Typically, the percentage of responsive cell in the field of view is quite consistent across animal. Although the number of cells we were able to image sometimes can be variable, depending on, because the surgery is kind of not that easy. Implant a big length into a different structure. We typically can image between 30 cells, lucky we can get 100 cells. 100 cells probably is the upper limit of our ability to do image. So there is a variability among, in the number of cells we image, but among those cells, typically there are 20-30% of cells will respond to a kind of UF, and only 1 or 2 cells respond to any sound, or no cell respond to sound at all. And those are pretty consistent across the animal. Yeah.
Thank you.
Can I just ask a follow-up question to that? Because the individual differences issue is a really important issue for psychopathology.
Hey, Richie, you got to speak up a little bit. Yeah.
Since we know that when people are exposed to adversity, there is huge variation in how people respond, which in many ways affects one's vulnerability to psychopathology. And looking at some of the learning curves that you presented, where you're looking at the slope before learning and then after learning, it looked, I mean, even though there's a very small number of animals, at least in some of the experiments, there's a lot of variation in the slope. And I wonder if you can envision a strategy for screening animals, potentially wild type animals, to look at individual differences in this way and potentially select those who, because of particular characteristics of learning, may have some vulnerability that can then be explored using the strategies that you've used.
Indeed, that's an excellent suggestion. There are huge variability when we look at the behavior. Indeed, in the learning curve, even fear conditioning, we're talking about variability between 30% to 80% of freezing after simple fear conditioning. I think some of the neuronal population that we study can contribute to the variability. Yeah, I think depending on the questions, we can segregate based on behavior to population, more than 2 population for animals, and ask whether some of the variability can be explained by the neuronal activity in the central amygdala. I think many of the behavior maybe can be explained by that. The anxiety temperament that just mentioned is very interesting. That you see this in a human primate, and you have a correlation between that and the expression level of Piccadilly protein expression, right? That's very striking to me, very interesting. I think that when I read that paper, I thought, okay, we probably need to do something, to look at it in mice. There's no good way to look at similarly in monkey compared to monkey in terms of the anxiety temperament, but we can use some assay in rodents to look at more or less anxiety related behavior in mice to segregate them and ask whether the Piccadilly cell really look different in terms of the behavior, in terms of the response to different stimuli. That is something that actually I'm very interested in doing. Yeah.
Great.
Maybe we'll take one more question. Take a break.
Hi, my name is Ceci and I'm a postdoc at Emory University. So my question is with regards to habituation. So I know that we've been talking a lot about saliency, but I was kind of surprised when you showed data a little earlier on in your talk where you showed that there was similar responses in these neurons across different days. It was 5 days to the different stimuli. And I was wondering whether or not you've collected data that looks at different, whether or not the neurons similarly respond across multiple trials of the same stimuli.
Oh, whether across files there is some habituation
Correct,
in
yeah.
response. Yeah, indeed. If we give repeated stimuli like 35 or so, if you look at the first several trials compared to the last several trials, there is habituation normally in the same imaging session. Yeah.
Okay, thank you so much both for a great session.
Thank you. Thank you. Thank you. Thank you very much.